Welcome back, friends. In this video tutorial, we'll be learning the T4 bacteriophage life cycle and the infectivity or infection properties of T4 phage. Okay, and in previous video, we have already talked about the T4 bacteriophage structure majorly. And usually, when we draw structures of bacteriophages, we always think of a particular structure which is having a head and a collar and tail and having the base plate and spikes are coming out. That's a typical representation of T4 bacteriophage. Okay, so let's begin this process. Now, let's first see the different stages of this T4 phage infection. And the different stages can be divided into sequential events. Events like first is absorption of bacteriophage to the host cell surface. And second thing is then penetration of that host cell, and so that actually when we talk about penetration, actually in this case viral particle, the whole body is not penetrating the host cell, only the genome of the phage is just ingested or injected inside the host cell. Now the third thing is the transcription of that injected DNA inside the host cell by capturing or hijacking all the other replication and transcription and translation machineries of the host cell. Then the replication of this genome, and then once we get larger copies and many copy of our desired phage genome inside the host cell, along with the necessary proteins to be packaged to package that genome inside, we are ready to go, and the phage particles will start the assembly, and they start assembly. After the assembly is done, they will be released outside by creating the burst or lysing the host cell membrane. Okay, so this is the sequential stages of how T4 life cycle actually occurs. Okay, so let's talk about each of these different stages in detail in the future slides. So if you look at the timing of the life cycle of the T4 phage, what we can find that at the 0th time towards the 22 minutes of the life cycle of T4 phage, there are sequential events that must be established prior to get a perfect infection. Now at T0, the adsorption of the phage particles to the bacterial cell achieved. In the T1 stage, synthesis of host DNA, RNA proteins are turned off. This is very important prior to synthesizing new mRNA for themselves. So they need to shut down the host DNA, RNA and protein synthesis. Now the T2 time, it synthesizes the first mRNA, and T3, degradation of the bacterial DNA occurs. After that, at T5, initiation of phage DNA synthesis begins, and after that at T9, synthesis of late mRNAs which are required for the synthesis of structural proteins are being made. After these structural protein mRNAs are being produced, translating them we get the structural DNA product. So at T12, appearance of complete head and tail and assembly of genome start to occur. At T15, appearance of first complete phage particle, and then at T22, lysis of bacteria and release of progeny phages. So we can see from the 0th minute to the 22 minute or 22nd minute, there are sequential events that are going on to finally establish what we call a phage infection inside the bacterial cell. Okay, so first let's talk about the adsorption and entry of T4 bacteriophage inside the host cell. Now remember, whenever we're talking about entry of T4 bacteriophage inside the host bacteria, we are talking about the entry of its DNA, not the whole phage. Now in this case, T4 uses E. coli cell wall proteins and polysaccharides as receptors. So the receptor mediated interaction with the surface of E. coli helps them to be attached to the bacterial cell surface. Then the tail fibers contact the appropriate receptor site, and then the base plate settles down onto the surface, and using the base hooks and the spikes, they are very much eager to hold on to the surface of bacterial cell. Now then the conformational changes occur in the base plate and the seed, and the seeds start to contract, and as we know, this T4 phages are contractile bacteriophages. That means they can contract their sheath. They are acting like a small syringe to deliver the DNA inside the host cell. So in this case, as we can see, the seed contracts due to the conformational change, and it delivers the DNA inside the host cell. Now usually the T4 infects only two different E. coli strains. One is E. coli K12, another one is E. coli B. Now the central tube or core is pushed through the bacterial wall. Now the base plate contains the lysozyme-like protein, which is termed as GP5, which aids in the penetration of the tube through the peptidoglycan layer. So here we can see the bacteriophage having this contractile tail region. Now as it is injecting, as it is pressing it down, it just starts to inject the DNA inside the host cell. Now once the phage DNA inside the host cell it will begin its activity. The DNA is finally extracted from the head and through the tail tube into the host cell is done. And usually for establishing this task, actually there are enzymes like GP5, as I've already talked. They behave like lysozymes so that they can degrade the bacterial cell wall components. Now let's talk about the second stage, or taking over the host cell machinery, which is very important to keep this phage functional for the further time. Now in this case we will be looking at two different scenes. One is synthesis of early proteins, then the synthesis of late proteins. There are also other certain type of proteins called the middle proteins, but we will not be talking about them in detail. Now in synthesis of early proteins, what we can see, 39 early promoters are present in T4 genome. Now the T4 genes are transcribed by the regular host RNAP and sigma factor. Now here GPALT is a type of protein which enters the host with the infecting DNA, and ADP ribosylates the alpha subunit of RNAP. Now in this case, this inhibits the transcription of the host genes and promotes the viral early gene expression. That's a very important task. After that, a short interval of two viral enzymes are coded. One is mod A, another one is mod B. And this mod A and mod B expression, which catalyzes the ADP ribosylation of both the alpha subunits of RNAP or RNA polymerase. Now, when you're talking about RNAP, we're talking about RNA polymerase, right? Because RNA polymerase are having two different sections. One is alpha subunit, another one is beta, and also sigma factor is there. Now here alpha subunit is being ribosylated in this case by mod A and mod B. Now this turns off some of the early T4 genes. Now the two viral endonucleases GP10A and 10B degrades the bacterial genome into short double-stranded DNA molecules, which are further degraded into smaller pieces by viral exonucleases. Because as the DNA of bacteria is becoming linear after the degradation by den A and den B, further exonuclease can act on it and break it down. Two early proteins, HEIA and MOT-A, modifies the RNA polymerase and stimulates the transcription of early, late, or middle genes. Okay, so let's talk about them. Now, in this case, in the activity of early, late, or middle genes, the MHC present in the T4 DNA is synthesized by two virus-coded genes. Now, those genes are really important. In this case, one is T4 hydroxymethylase and HM kinase. Now, in this case, T4 hydroxymethylase methylates this cyclic monophosphate of DCMP into DHMP, and then HM kinase will act on DHMP to convert them into DHDP, because this monophosphate now is being added one phosphate by this HM kinase, and it is converted into H monophosphate to H diphosphate. In this case, inhibition of entry of cytosine. Now in this case, this DCTP or DCDP can be taken, and DCTase is an enzyme which cleaves the additional phosphate from cytosine and convert them into DCMP, which is no longer functional in that case. Now following the T4 DNA synthesis, HMC is glycosylated by two enzymes, and the enzymes are alpha glycosyl transferase and beta glucosyl transferase. So both of these enzymes are required. Now this glucosylated HMC protects the T4 DNA from attack by E. coli restriction endonucleases. That's why this modification is important. So in a nutshell, what they are doing, first of all, they are synthesizing their proteins, some important factors that are helping them to degrade the bacterial DNA, and then before the DNA replication begins, they start to modify the bases, because we know that in T4 bacteriophage there are modified bases present. So that's how they modify their bases, using their own enzymes called T4 hydroxymethylase and HM kinase. Now using them, they modifies there, and after the modification they replicate it and transcribe it, and after that when the T4 synthesizes the HMC and it is done, in this case they start synthesizing alpha glucosyl transferase and beta glucosyl transferase, which further blocks the activity of E. coli restriction endonucleases onto the T4 DNA, T4 Phage DNA. Okay. Now let's talk about a little bit of the synthesis of late proteins, which just came after the synthesis of early and middle proteins. Now in this case, 50 late promoters have been found in T4 genome, right? And in this case, in all of them, two important factors are very important, which is completely phage encoded factor. I repeat, phage encoded factor, and the factors are GP33 factor, GP55. So it is one is activator, which is GP33, and GP55 is a kind of sigma factor. which replaces the host sigma 70, because we know if sigma 70 remains there for longer period of time, then it will behave like normal host cells, because now they try to remove all the host cell like attitudes, and all one they want is to restore their own attitude there. So they are removing all the sigma 70s with GP55, which is having another sigma factor like activity. Now, late transcription is responsible for the synthesis of mostly structural genes like T4 head, tail, fiber proteins, virion assembly factors, and recombination proteins. Because we are going to see these recombination proteins are going to be required for the replication process in a little bit amount. Now, let us talk about the replication of T4. Once after they achieved, they hijack the machinery of host cell, and they start to make early proteins and late early proteins or middle proteins. In this kind of condition, they need to duplicate their genome. Otherwise, they won't package something, right? Because for the packaging, they require genome as well as the structural parts. They may produce structural parts from the previously produced mRNA transcripts, but for the packaging of the DNA, they require to produce this genome. Now, the T4 DNA replication requires several viral coded proteins. One is T4 DNA polymerase, primase, helicase, and recombination exonuclease, single strand binding protein, etc. Now, the T4 DNA replicates in two phases. One is which is called the first phase of replication. Then we will be seeing the second phase of replication, which is slightly modified. In the first phase of replication, it begins at multiple origin, just like a formation of replication fork or replication bubble, which will migrate to both these directions, and finally they will meet at a particular location. So that's how they can begin this process. But during this process, they encounter a problem. Now, what is the problem? If we are talking about linear DNA, in that case, if we are talking about linear DNA just like the DNA of T4 virus, now in this case, as the DNA is linear, each time when they are adding primer at the terminal ends, and they start synthesizing all the DNA in both the strands, usually in the opposite or lagging strand, we need to add primers, right, for the synthesis, because the DNA polymerase can only synthesize from 5' to 3' direction. So here in this picture we can see red things are the primers that are being added. After the addition of primer, then rest of the nucleotides are being added by the DNA polymerase, and the polymerization is going on. But what is the problem is that each time after the synthesis is complete, they need to remove this DNA primer, because the primer is made up with RNA. So I must call it RNA primer except for DNA primer, because it is made up with RNA or ribonucleotide segments. So this RNA must be removed from this DNA, total DNA backbone. So for that reason, after the synthesis, they need to chew these RNA strands apart. So if they chew these RNA strands apart, as a result of that, what we get a terminal gap in both this case after each replication cycle. Now that creates the problem, because if it is going on and on and on, just like what happens in case of eukaryotic chromosome, the end part is kind of getting lost each time it takes a replication. So it is very difficult, because the information is being lost, genes are going away, so they won't tolerate that. So to prevent that, what they can do, there are two different solutions for that. One type of viruses, some type of viruses just do it, they make a concatemer formation. As a result of concatemer formation, what they can produce, the cos side or cohesive ends, right? Just like the lambda phage, they produces cohesive ends in that case. So as they are making cohesive ends, they can easily circularize their DNA once they inject their DNA inside the host cell. That's in case of bacteriophage lambda. But in case of bacteriophage T4 and T7, how to remove that kind of condition? They can remove it via the formation of concatemers, and again similarly via the formation of terminally redundant sequence. Now, what do we mean by terminally redundant sequence? We have already talked it in a structure of genome of T4 phage in the previous video. If you remember or recollect the material, the terminal redundancy means at both the ends we are having same type of genes. Just let's begin with 1, 2, 3, 4, 5, 6, and after this 6 gene we are having again 1, 2, 3. Just so what we are having at both the terminal, we are having same genes repeats in both these cases. So this kind of terminal redundancy can help them to block this kind of gene loss effect. Now the second phase of replication is the genetic recombination that can proceed within the regions of terminal redundancy of two daughter molecules. And the second phase of replication is unique to T4 phage, because in phage like lambda they won't have any terminal redundancy in those cases, because they just have the cos sites that is produced during the packaging of the phage DNA inside the head. But in this case, what we can see, in this case the terminal redundancy, as they are having the terminal redundancy, as you can see here, A B C D E F, and then again W X Y Z, after the Z again A B C D. So it's kind of continuous repeat of same sequences, and at the beginning and at the end, both this case we are having same sequence A B C repeats. It may be repeated for 1,000 times, it may be repeated for 500 times, but it should be repeated in both these ways and correctly. Okay, so as they are having this kind of redundancy, they can kind of having a recombination between themselves. So as you can see, as they are having a recombination, we can attach two different gene segments like that, and this is important. This kind of recombination take place during the second phase of replication of T4 phage, and it gives rise to molecular concatenamers. So as they are giving rise to concatenamers, means a long stretch of DNA having same sequences attached all the time. So that means this from A to this Z is necessary gene for to be encoded and to be packaged inside a head, and from this A to this Z again is ready to be packaged in another head. So they are forming this kind of long similarly attached genome, multiple genome attachment. So then they will cleave this each genome and they will package this genome inside the host cell, or inside, not host cell, inside the bacteriophage head. Okay. So here we can see the recombination, which dependent and replication dependent upon the recombination. Now in this case, there are two models. One is the bubble migration synthesis model. Another one is the Mosing model of T4 replication recombination, or terminal redundancy, using terminal redundancy. Now in this case we can see that this is a strand and this is another, which is having a kind of deleted part, which is a newly synthesized. Now in this case it will be strand invasion in the first place. After the invasion, it starts replicating in both the ends. After the replication is done, branch migration and the resolution of this holiday junction, and what we get is two different genes. But above all, in this both the genes, this thing is possible because they are having similar sequences in both this DNA. If they are having different sequences, this thing may not be possible, right? In case of bubble migration system, in this case also we can see strand invasion will be there, initiation of synthesis will be there, but synthesis will only occur in one particular part. It will occur in synthesizing at front and branch migrate at back, right? So in both this case, synthesis will occur like that. After the synthesis is done, it continuously synthesizing the whole strand and it will be apart. And after that again the other strand will invade and that will synthesize their own way. So that's how the two models are explaining how the recombination can take place during this second phase of replication of T4 phage. Now let's talk about the assembly of T4 bacteriophages. Now before assembly, they must produce all the different proteins required for the assembly. Proteins majorly important as called capsid proteins, called accessory proteins, and called scaffolding proteins. Now these three proteins are major and most important proteins for giving the structural property to the bacteriophage. And we obviously require connector protein, proteases, and terminase to help the process to guide. Okay, so in this case, in the very beginning, in the formation of procapsid, it requires three proteins: capsid proteins, which will be arranged outside, then the accessory and scaffolding proteins. Now, scaffolding proteins will guide these accessory proteins to be attached with each other to make an inner core, and then these capsid proteins will be added in the outer core. Once they produce this kind of arrangement, we will be calling it as a procapsid, and it will be having a small pore-like structure here, which is called connector. Now, this connector is really important. Now, as the capsid protein proteolized and remove the delta domains from them, and then connector will come here. So scaffold proteins must be removed because they are not the part of structural integrity. They are only helping this organization. So then they will be proteolized using protease enzymes and they will be cut away. Then what we get is a procapsid, which is a processed procapsids after cutting the scaffolding out. Now in this processed procapsid, there is a channel formed using connector molecule or connector protein segment. Now at this particular procapsid state, it begins the packaging of DNA. Now as the DNA packaging will be going on, this head will be matured more. Now as it is going on in this case like that, then it will bring this DNA, and the guide molecule is also important in this case, and the guide protein for pumping the DNA inside the head will be added by this by this terminus proteins, which is having a helicase like activity. It will wrap and move this DNA very fast using the energy getting from ATP hydrolysis in this case, and it will be pumping the DNA inside. Once the DNA is being pumped inside, during that point they get more and more accessory proteins coming in, and they will be added to the outer membrane, or what we call as capsid proteins from outside, and the head is being matured and more. And at this particular place, when every DNA is being inserted, it will be packaged properly, it is ready to attach to the tail and fibers, and then it will be called as a matured head. Now the head full of DNA is 3% longer than the genome of actual genome size, because this is due to the terminal redundancy, which is simply the permuted molecules with repetitious ends in both this direction. So 1 to 3 and end with again 1 to 3, similar like that. So for that reason, it will be packaged slightly extra, which is 3% extra inside, right? Now the tail assembly. Now the tail and base plate assemble separately. Now the base plate is constructed of 15 gene products, and then after the base plate assembly, the tail fibers are next attached to the base plates, and the tube tail and the tail tube is built on the base plate. So you can see base plate is the base. That's why it's called the base plate, because base plate is the first thing. Then the base fibers are attached to it. Then the tail tube is start to build onto this base plate, and as the base plate and tail tube is built, they will come and they will be attached to this viral head. After this attachment, the seed is assembled and around the tail tube. That's how the tail is assembled. Once the tail is totally and completely assembled, I mentioned it wrong. Once the tail is completely assembled, the tail will be there, and it will be directly and randomly attached with the phage head. Now this attachment of phage head with the tail is a kind of random. Nothing can be predictable in this case. Now once they have attached this head with these tail particles, they produce these mature phage particles or virion, which can be infecting, right? Which is getting the infectivity property from now on. And once they reach a particular threshold of phage inside the bacterial host, they will burst the cell open and release outside. Usually in this case of T4 phage, the burst size is 300. That means when they reach 300 or more phage particles inside the E. coli cell, they will kill the cell and will be released outside. So release of new T4 bacteriophages from the host. Now in this case, the lysis of host E. coli cells take place after about 22 minutes at 37 degrees Celsius temperature, when approximately 300 T4 particles are released. Now the T4 and many other phages lyse their host by damaging the cell membrane and cell wall. Now in this case, two enzymes stood still for this to be done. One is choline, another one is lysine. Now choline is a T gene product, which is an enzyme which destabilizes the host cell membrane. That means it creates a hole inside the membrane of E. coli. And the second thing is lysine, which is a phage enzyme which breaks host cell wall. That means it lyses the host bacteria, and then after the lysis, so that the phage particle can come outside. Okay. So that's the process, and using these enzymes they create the hole and burst the cell, and all the phage particle will be outside. But definitely they require some environmental factors. Obviously the time is almost 22 minutes to complete the whole process that I've discussed. It took only 22 minutes for the phage to complete at 37 degrees Celsius for as optimum temperature, because that's the temperature that we get in human body, and then our bacterial cells, our bacterial optimum temperature, and approximately when they are having 300 T4 particles, right? So that's the process of how T4 phage infects the bacteria and kill it, and they replicate themselves inside. And I hope that's helpful. Thank you.